So here's a lateral view of the skull, and some of the bones have been sectioned through so that we can see the cranial cavity and the orbit. So let's name some of these things. So which muscle is this? Good, the superior rectus. What about this one? Good, lateral rectus. What about this one? Good, inferior oblique. What about this one? Good, inferior rectus.
Is it inferior oblique rectus or just inferior oblique muscle?
Oh, inferior oblique muscle. Yep. Mm-hmm. Okay, good. We don't see it, but what's the one like over here?
Superior oblique.
Not superior oblique, but good, medial rectus. Good. Because superior oblique is right here, right? Okay. Okay, so those are the extraocular muscles. And some of the cranial nerves innervate these extraocular muscles, right? Some of the functions of the cranial nerves are eye muscle control or eye muscle movement, right? But let's go into specifically like which cranial nerve innervates which eye muscle, right? So we can just follow the nerves, but let's try to identify them first, right? What is this? Sorry, like which part of the brain is this?
This is midbrain.
Good. Here? Good. Here? Okay. Good. So it's like, okay, here's midbrain, here's pons. So this is like superior to the pons, and there's, right? Oh, sorry, which one? This one or this one? Because both of those nerves are superior to the pons.
The one to the left.
This one is oculomotor?
No, this one is trochlear.
It's got to be trochlear, right? Why is it trochlear?
Because it's kind of hugs over on the side, like on the back part of it.
Right, it's more lateral, right? So like here, like right here, superior to the pons, but lateral to the midbrain is always the trochlear nerve, right? So it doesn't matter if you're looking at it anteriorly or laterally or posteriorly, that's its relationship to the midbrain and the pons. The fourth cranial nerve comes out lateral to the midbrain and superior to the pons, right? Okay, what about this one?
Oculomotor.
Good, oculomotor. Where does the oculomotor nerve emerge?
It's kind of like from the superior portion of the pons.
Right? But so does the fourth one. So how do we differentiate them?
No, the fourth one is hugs on the lateral side behind it, and that the oculomotor kind of like over on the top, over the pons.
But they're both over the pons. How about oculomotor is more medial,
Okay, more medial.
right?
There you go.
And it's more medial to like which structures? Like which structure of the midbrain is this specifically?
The peduncle.
Which peduncle?
Cerebral.
Right, the cerebral peduncle. Good, right? So the fourth nerve emerges lateral to cerebral peduncle, but the third one emerges medial to the cerebral peduncle, right? Those are relationships that you should memorize. Yep. Okay? Like what is this one?
Trigeminal.
Good, the trigeminal nerve. And like where is it? Like describe it relative to something else.
It's lateral on both, bilateral on pons.
Okay? So it's like on the lateral pons, like in the middle of the pons, right? It's not like superior pons or inferior pons. It's like right in the middle, right? So lateral and in the middle of the pons versus, what about this one?
This one is inferior medial.
Okay, so it's like inferior to the pons, superior to the medulla. It's more medial. Which cranial nerve?
Abducens.
Good, the abducens nerve. Good. Okay, and then what is this?
This must be
Think of like the circle of Willis. This artery becomes the middle cerebral artery. Good. Must be the internal carotid artery. Nice. Good job. Good. Okay, so yeah, anatomy is spatial relationships. Like, where is this thing relative to something else? And then you can just take those words, you can apply them to anything, any diagram, cadaver, prosection, whatever, right? Okay. Here, let's just follow it. So here's oculomotor. Which number is it?
3.
Good, 3. And then it goes in here, and then you can see that it's going to innervate this muscle, which we don't learn about, this muscle, which we do learn about. What is this muscle again? Good, superior rectus. This one, which one is this? Good, inferior oblique. It does this one, which one is this? Good, inferior rectus. And it's showing like this branch going more medially. So which, good, medial rectus. Good. So the oculomotor nerve innervates 4 of the extraocular muscles: superior rectus, inferior rectus, medial rectus, and inferior oblique, right? So that means that there's 2 more muscles to innervate, and there's 2 cranial nerves. So each cranial nerve innervates 1 muscle. So let's follow this one. Which one is this again? Good, trochlear. So here it is, and it goes, good, right? So it innervates the superior oblique muscle. Good. And what about the last one? Good, abducens. What does it innervate? Good, the lateral rectus. So it goes in, and then you can see some of the branches here, right? Innervating lateral rectus. Good. Okay, so let's talk about what's the function of the abducens nerve specifically then? Eye muscle control, sure, but like how does it move the eye? Good, it abducts the eye. That's why it's called the abducens nerve. Good. What about the function for the trochlear nerve?
Eye muscle movement.
Okay, good. Eye muscle movement. And specifically, since it innervates the superior oblique muscle, it causes your eye to depress and abduct. My professor described it as the cheater's nerve. Like if you're taking an exam and somebody's next to you and like you don't want to like get caught cheating, you just like kind of look like that, right? Because that's what the trochlear nerve does. It does depression and abduction of the eye, right? So that's the specific eye muscle movement that it does. Okay, and then what about the oculomotor nerve? We can just say all other movements of the eye, right? Elevation, adduction, yes. Like all of the other axes, yes. Good. I just think it's helpful to like know specifically that these nerves are going to specific muscles, and each muscle does a different thing. So you can still say eye muscle movement, eye muscle control, but like this is how these cranial nerves are controlling the eye, right? By innervating specific extraocular muscles. Okay, good. All right, here's a superior view of the orbit. Let's name some structures. So, what is this?
Superior rectus.
Good, superior rectus. What is this? Here, go ahead and look at, it looks like it's going here, and then it goes through that, and then it does that.
Oh, that would be the superior oblique.
Good, superior oblique. What about this one?
Inferior rectus.
Good, inferior rectus.
This is a long view.
It's a superior view. What about this one?
That's inferior oblique.
Good, inferior oblique. It's also over here. Yeah, because it attaches to the, I don't know which bone that is, but some bone of the orbit here, and then also to the sclera. Okay, what about this one?
Medial rectus.
It's got to be medial rectus. Good. Okay, what about here?
Lateral rectus.
Good, lateral rectus.
oblique muscles are kind of shaped like boomerangs.
Yeah. Mm-hmm. Good.
Because they are responsible for rotation too.
When you say rotation, do you mean like
Eyeball rotation.
like the intorsion and extorsion of the eye?
Yeah.
Yes, good. Did you learn about that? Intorsion, extorsion?
No.
No.
It's like when you rotate your head, the image stays still up to a certain point, and then it starts to rotate. Because when your head rotates, your eye like counter rotates. So it's called either intorsion or extorsion. And that's what those superior and inferior oblique muscles do, right? They do intorsion, extorsion up to a certain point. I don't think we have to know that unless your instructor mentioned it. Okay, good. Okay, what about this one? Like, what is this? Good, the optic nerve. Nice, good. All right, okay, good. Oh, also, I think it's useful to know this structure. It's called the trochlea. Trochlea means pulley. You remember the trochlea and the humerus? So this is another trochlea. So they named the trochlear nerve because it innervates the superior oblique muscle, and the superior oblique muscle goes through the trochlea. So that's why they named it the trochlear nerve. All right. Okay, so here's the eyeball, the globe. There's lots of names for it. This is a superior view and a transverse section. So let's talk about what is this, like all of this space.
Anterior cavity.
Good, the anterior cavity. Let's see if I can. Okay, good. And the anterior cavity has two more spaces. What is this space?
Anterior chamber.
Good, the anterior chamber. What about this space?
Posterior chamber.
Good, the posterior chamber. Good. Which fluid is within the anterior cavity?
It's aqueous.
Good.
Aqueous.
Aqueous humor. Good. I don't know if you can call it aqueous fluid. Yeah, aqueous humor. What does aqueous humor do? Okay, good. So it provides intraocular pressure to maintain the shape of the cornea. Good. What else does it do? Good. So it's there to facilitate nutrient and waste exchange for the cornea, because the cornea is avascular. There's no blood vessels for the cornea, but there's cells in there. So how do those cells not die? Through the aqueous humor. Good. Okay. Yeah, what is this? Good, the cornea. What's the cornea do?
It bends the light rays.
Good, right? It bends the light. What's the word for that? Good, good. It refracts the light. Good. Yeah, most of the, I forget what it's called, like focusing power, refraction power is because of the cornea, right? There's all these structures that refract the light, right? This refracts and that refracts, and even these like slightly refract, but this one does the most refraction, the cornea. Good. The cornea also protects against like microorganisms. It's like an actual physical barrier. Okay, good. What is this? Good, the iris. What does the iris do? Good, right? Controls the diameter of the pupil. Good. What does the pupil do? Exactly, lets light in, right? It's a hole. Good, good. Okay, good. What is this?
Lens.
Good, the lens. What does the lens do? Okay, good. So the lens can change shape in order to focus on near or distant objects. Good. The lens also refracts the light. Yes. Okay. And okay, let's talk more about lens accommodation. Which cranial nerve is responsible for lens accommodation? Good, the oculomotor nerve. Good. Yeah, the question was, which cranial nerve is responsible for lens accommodation? The third cranial nerve, the oculomotor nerve. How does it do that? By innervating like which muscle? Right. Good. Good. The ciliary muscle. Good. Within the ciliary body. Good. Good job. Yeah, and the lens, its normal shape is a sphere. Like it wants to assume the shape of a sphere. And sphere is for near vision. So if you're looking at something like close to you, your lens is shaped like a sphere, versus if you're looking at something far away, it's more flat. So flat is far, but sphere is near. Okay, so if that's the case, is the oculomotor nerve sending sympathetic or parasympathetic fibers to innervate the ciliary body? Good, parasympathetic. And when it does that allow you to see things that are close or far away?
When it contracts or relaxes?
Close,
Close?
right? Yep.
Oh, then it's, I forgot the question again.
Sorry, let me explain it. So it's like we're trying to figure out, okay, oculomotor, parasympathetic. Parasympathetic is rest, digest. It's not fight or flight.
Right.
So it's like, it's more useful to fight and flee. Like it's more useful to have like far vision if you're trying to fight and flee, right? Because you can see the threat from far away versus like if it's like right here, it's too late. So that means that the oculomotor nerve is not doing far vision. It's not making the lens flat. It's making it more spherical. So it's causing the ciliary body to contract. And when the ciliary body contracts, that makes these structures more slack, and then this becomes more like a sphere so that you can see near, right? So all of that goes together. It's like, okay, sphere is near, it's parasympathetic. How is it parasympathetic? Because you're seeing things that are close, not things that are far, and when the muscle contracts, it doesn't cause the lens to flatten out, it causes the lens to become more like a sphere, right? Okay. Okay, and then what are these?
Suspensory ligaments?
Good, the suspensory ligaments. I think they call them, that's another name for them, but just call them suspensory ligaments. Okay. What do the suspensory ligaments do?
Transmit tension and enable the lens to change the shape.
Exactly, good, that's great. It transmits tension into the lens, allowing it to change shape. Exactly, good. All right, what is this? Good, the ciliary body, good. Yeah, and we're not distinguishing between the ciliary processes and the ciliary muscle. I don't remember what it is, but the important thing is that all of this is smooth muscle, right? You can call the whole thing the ciliary body. What is it made of? Smooth muscle. What kind of smooth muscle? Or like how should you describe this smooth muscle? It's ciliary muscle, okay? So call it ciliary body, the whole thing, because I don't think we're differentiating between the processes and the muscle. In lab, at least, maybe in lecture you do. Okay, what about here? Good, the canal of Schlemm. Exactly.
What's that name?
We have another name we get to use.
Yeah, it's also called the scleral venous sinus. Scleral or sclero, something like that. Scleral venous sinus. Say it again. Yeah, it's the.
Yeah, it's scleral
Scl-
venous sinus.
There we go. Scleral venous sinus. That's another name for it. What does it do?
Pressure.
Okay, good.
Lens aqueous humor.
Good, right? Drains aqueous humor, relieves the intraocular pressure of the eye. Exactly, good. What is that disease called if there's too much intraocular pressure? Starts with a G.
Kind of
Like when you go.
Starts with a G.
Mhm. Oh.
What the hell? My chair, but I didn't do anything.
Mhm.
My fault.
Glaucoma.
Good, glaucoma. Good, glaucoma. Like you ever go to the eye doctor and they do like the puff of like air into your eye? What they're doing is they're testing for glaucoma because glaucoma is increased intraocular pressure. So they're like blowing the air onto your cornea and there's like a camera here and they're watching to see how much does your cornea move, because if it doesn't move very much, that means that there's too much aqueous humor. So that means that you might have glaucoma. And maybe that means that there's like a blockage in the scleral venous sinus. Or maybe it means that the structure producing the aqueous humor is producing too much of it. What is the structure that produces aqueous humor?
Chlorine.
Good, right? The ciliary processes of the ciliary body.
There's a change.
Okay. Okay, good.
So wait, we're trying this aqueous humor back to the blood?
Mm? Yeah. Yeah, back into a vein.
most of the liquids, right?
Mm? Good. And is it in the lab manual, ciliary process? Or is it just ciliary body?
Ciliary body, no need to distinguish the ciliary process and ciliary muscle.
Okay, yeah, there you go, right? So just call the whole thing the ciliary body. There are like muscles, the ciliary muscle, and then the processes is a different like structure technically. Okay. Yeah, and this canal of Schlemm is always here at this, I forget, this angle in between the cornea and the iris, right? So if there's anything tagged here and it's a hole, it must be that. Or if you're trying to find it on a model, like it must be here, like at this iridio-corneal angle. Okay, what about this?
Oh,
That's membrane.
It is a membrane, yes.
Is that supposed to be the conjunctiva?
Good, the conjunctiva, good. They call it the ocular conjunctiva because this is the part like on the sclera, but then there's also a part on the eyelid. So I don't think we have to differentiate. All of it is the conjunctiva, but just know that it's on the eyelid and on the eyeball itself. What's the function of the conjunctiva?
protection.
Okay, good. Protection against what?
Protect, lubricate, and reduce.
Okay, good. So like
Mucus.
So like protection against like microbes or particles from entering like into the orbit, right? So like a physical barrier. Protection against desiccation, right? Because it's lining the eyelid, and every time you blink, you're washing like the fluid from the lacrimal gland over all of the cornea and the sclera. So it's also like preventing the eye from drying out. Okay, good, right?
And stratified squares.
I don't remember actually. Yeah, I don't remember. I don't think it is stratified squamous. I think it transitions. I think it's complicated.
It says, cuboidal, stratified cuboidal,
Hmm.
and our teacher said it's more of the stratified squamous than it is cuboidal. I don't know if it's important for the lab test.
Yeah, no, I don't think so. I think it's complicated though. I think it like transitions from cuboidal to squamous. Simple cuboidal, I don't remember. Yeah, I don't remember. Reference a textbook, yes, yes. Okay. All right, let's keep going. All right, what is this space?
Posterior cavity.
Okay, the posterior cavity. And then what is this within the posterior cavity?
Vitreous.
Good, vitreous humor, good. Or the vitreous body. What does the vitreous humor do?
Maintain our shape.
Good. Maintains the shape of the eye. Good. Okay. What about, which muscle is this? And this is a,
Lateral rectus.
why is it the lateral rectus and not the medial rectus?
where the ocular nerve is.
Okay. Okay, what about the optic nerve?
It's coming in from the side.
Um,
Coming in more medial.
right, it's like, so the optic nerve is more medial, like it faces medially, right? It leaves the eye going medially, right? So this must be medial. This must be lateral. So this must be the lateral rectus. This one is what? Good, the medial rectus. Good. Yeah, because that's the only way we can tell, right? Because otherwise, like without that, there's no way to tell, like, is that medial or lateral? It's like perfectly symmetric. But then you see the optic nerve, and the optic nerve goes medially, right? So this must be medial, okay? Which layer is this?
The sclera.
Good,
Sclera.
good, the sclera, good. What's the function of the sclera?
Connective tissue.
Okay, good. It's connective tissue. It maintains the shape of the eye. Yes, good. Nice, good, good job. What is this layer? Good, the choroid. What does the choroid do? Right, so provides metabolic support to the retina. Good. What else does it do? There's another important function. Like it's vascular and blood has some color to it, but then there's also pigment within the choroid. Why?
Oh, are we talking about the pigment that is like on the last layer?
You can think of that
It
too.
absorbs the light.
Good, right? So absorbs excess light to prevent scattering of light, right? So like when the light comes in, you don't want the light to like bounce off and then like activate this and then like bounce off and then activate that, right? You want it to like go and activate that one part of the retina and that's it, right? So like the choroid, along with the retinal pigmented epithelium, can absorb excess light to prevent the scattering of light within the eye. Good.
Can I ask you a question? Is that
Mhm.
because I think I read something that melanocytes are responsible for that. Is that?
Mm-hmm. Yes, good. All right. What is this layer?
The retina.
Good, the retina. Good. What does the retina do?
It has like it's like the neural layer.
Neuroreceptors.
Mm-hmm.
It's like the cones and rods.
Okay, what do cones and rods do?
They perceive color for the cones, and then I forget for rods.
Rods are dark and light shades,
Okay, good, right.
black and white, and cones is color,
Good,
color.
good, right. Let's summarize all of that and say the retina performs phototransduction. Phototransduction is taking light energy and converting it into electrical energy, I guess, electrochemical energy, like into the action potential, right? So that's what the retina does, right? There's two types of photoreceptors, the rods and the cones. The rods is for like black and white. The cones are for color. Yes, good.
Can you repeat that? Converts light into a chemical.
Into electrochemical energy.
Or performs phototransduction.
Mm-hmm. That's what phototransduction is, yes. Yeah, transduction, that word means like to take one energy and turn it into a different energy, right? What's another transduct? Like we're doing sound, right? So sound transduction, right? turning sound energy, which is like mechanical energy, into electrochemical energy too, right? All of our special sense organs are transducers, yes. Okay. And then within the retina, there's what's like this, like the more broader region?
Oh, that's the macula lutea?
Good, macula lutea, good. And then what about this specific part of macula lutea?
Fovea centralis.
Good, fovea centralis, good. What's the function of fovea centralis?
Central vision and the visual acuity.
Okay, good, right? So produces high resolution color central vision. Yes, good. That's what the entire macula lutea does, and then the fovea centralis does it the best. When we're seeing things, that's what we're doing. We're trying to place our fovea centralis on things, and we're using the cranial nerves to innervate the extraocular muscles to do that, right? That's the whole point. Like, this is the whole point of the eye, really. Like,
What's the
Mhm.
phrasing you use for fovea centralis?
macula lutea.
Or for the macula lutea?
Oh, sorry.
Visual acuity.
Oh, sorry. Say your question
I
again?
was asking what was the verbiage you used for?
Oh, the function?
Yeah.
Produces high resolution color vision, central vision. I don't remember what I said.
Central vision and visual acuity. I think that the best term is acuity, right, because it has high resolution.
Yeah, visual acuity, high resolution, that's the same thing. Yes.
I have a point of highest acuity.
Yes. Yes. Good.
How do you pronounce that word?
Acuity.
Oh,
Mm-hmm.
I'm just equity.
Equity
It's
is
priceless,
something else.
right?
Yeah, yeah, acuity, resolution, it's the same thing. It's like, 4K versus like 144p, right? High resolution, high acuity versus low resolution, low acuity, right? So it produces like high acuity central vision with color. Yeah.
Concentration of the cones.
Highest concentration of cones, contains highest concentration of cones. That's another good function. Yes, good. Yeah, devoid of rods. Yes. Okay, good. Which nerve is this?
Optic,
Good. Which number?
2,
Good. Function?
vision,
Good. Produces vision. Transmits visual information. Good. And then what is this? Good, the optic disc. What does the optic disc do? Or like, what is it?
It's a hole in the retina.
Okay, it's like a hole in the retina. Good.
it's a blind spot, right?
There's a blind spot here, yes. Why is there a blind spot? Because all of the axons from which cells are converging here? Which cells?
The ganglion cells?
Good, the ganglion cells. Good. What's another function of the optic disc? You can kind of see like these things here.
These are blood vessels, aren't they?
Mm-hmm.
So it just allows passageway for nerves, fibers, axons, blood vessels.
Exactly.
Just name it.
Good.
Doesn't
Yes.
have any cones or rods, so it's a blind spot.
Good. Yeah. So also allows blood vessels to enter and exit the eye. Like when they do I forget what it's called, ophthalmoscope, something like that. Like when they use that ophthalmoscope, they know that they're looking at the optic disc because they can see the blood vessels like going into or leaving the eye, right? So that's another function of the optic disc, is to allow for blood vessels to enter and exit the eye.
Oh, I had a question.
Mm-hmm.
My grandma has like macular degeneration, so
Mhm.
is it a problem with specifically the fovea centralis, or is it?
Yeah, it could be specifically fovea centralis, or it could just be like any one of these zones of the macula, because there's different regions of the macula, and so it could be like anything in there. Yeah. Mm-hmm. Okay. I don't remember why this one is in here. Let's just go through it though. So this is like a superior view, a horizontal section. What is this? Good, the cornea. Here. Good, the iris. Here. Not the sclera. Good, the conjunctiva. Good. Here. Good, ciliary body. This. The retina. Good. Here. That's the sclera. Good. Oops. Yeah, this has to be the sclera because the sclera is continuous with the cornea. What's the name of that entire tunic? Good. Good. The fibrous tunic. Good.
That's regular, right? On the cornea.
Yeah, this is dense irregular connective tissue, but the cornea is. I don't remember because there's cells in there too. Is it dense regular or dense irregular?
Dense regular.
Dense regular. Yeah, that sounds right.
Yes,
Yep.
that's because it needs to be clear and see through, so it's so extremely regular.
Mm-hmm. Okay. Yeah. 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 There's lots of layers of connective tissue. Yeah, you're right. Okay. Good. Good. What is that? I don't know. Let's see. Neural layer. I don't remember why this is in here. I think actually it was because we used to talk about the pigmented epithelium in lab, but not anymore. Okay, so don't worry about that. Yeah, the book talks about it, and maybe in lecture you also talk about it. Yes. All right, this is like a coronal section through the eye and a posterior view. So we're looking like. We're like in the posterior cavity looking at this crystalline structure. So what is this structure?
This is the lens.
Good, the lens. Good. So that's the lens. And then you'll see like what's anterior to the lens, like all that brown there.
That's iris.
Good, that's the iris. Good. What is the space? The pupil. Good. Oh, here. So here's iris. Where are they? What are these things?
These are the ligaments, suspensory ligaments.
Good, the suspensory ligaments. And what is all of this? Good, the ciliary body. Nice, good. Yeah, in lecture, I think you talk about the ciliary processes, so like these white things. Oh, no? Oh, some? Okay. Hit or miss, okay. Yeah, these ones are the ones that produce the aqueous humor. Okay. What is this? Good, the retina. And it's saying optical part because the retina is really like two parts, like the optical part with all the neurons, and then that layer with like the pigment, right? So that the light doesn't scatter. So that's what it means. What about here? The blue? Sorry, good, the choroid. What about here? Good, the sclera. Good, right? Good.
And between retina and choroid, there should be, is it between retina and choroid the pigmented layer?
Correct.
Okay. And choroid is the one that has all this vascular stuff.
Mm-hmm.
And the sclera is that dense irregular that holds the shape.
Correct.
Okay.
Nice. Good job. Good. Okay, good. Yeah, there's one model in the lab that looks just like this, except It doesn't show the suspensory ligaments, but it looks exactly like this, right? Okay. This is, I don't know, this is just for fun. Like, it just is a cool picture. Like, this is what your iris actually is, like a bunch of muscles. There's like a muscle arranged in like a circle, and then there's a muscle arranged in radially, yes. So this is how the diameter of the pupil is controlled, right? Through these two like types of muscles, the circular layer and like the radial layer. And then the iris is also pigmented. This is also to prevent light scattering. And people have different colored irises because our genes come from different parts of the world, and different parts of the world are exposed to different amounts of light and different amounts of ultraviolet radiation, right? Okay. What is this? That's the cornea. Good. Yeah, so pupillary sphincter, pupillary dilator. These are not in the lab manual. All this is just for fun. It's in the book, yes.
And pupillary dilator, I think we have it in the lab manual explaining the parasympathetic of ocular motor.
Oh, it says pupillary dilator?
I want to say so, but.
I think you're right. I think it does.
Oh, it says just a sphincter layer of iris constrict. Oh, never mind. This is parasympathetic. The sympathetic would be the pupillary.
Oh, yeah.
sphincter, right?
Yeah, okay, good. Yeah, it wouldn't be a dilator. It would be the, exactly, yes. Good, good. Yeah, because it's more useful to have lots of light coming into the eye in a fight or flight situation versus having less light, which means that dilator must be sympathetic and constrictor must be parasympathetic. Good? Okay.
Well, that's useful for dual innovation for the lecture. Well, I guess I'm the only one who didn't take lectures.
You haven't taken the lecture exam yet?
No.
Oh,
On Monday. I think everybody already did that.
okay. Got it, got it. Okay. All right, this is the same thing. I can just let you guys do that. Let's move on to like some of the histology. So here's retina. Here's like a schematic diagram cartoon of the retina. Let's do this one first, actually. So let's start here, even though this is not in the lab manual, but what is this layer? This is the pigmented layer, right? The retinal pigment epithelium. So this is, this thing is useful for preventing light scattering, and also this is where the photoreceptors like attach to the rest of the eyeball. Like you've heard of like a detached retina? Well, this is how it detaches. These photoreceptors leave the retinal pigmented epithelium. This layer also helps like the photoreceptors with metabolism, because they need to take light energy in and convert it, and that can produce a lot of like harmful waste products. And so this helps to deal with those harmful waste products too. Okay. But we don't have to know that for lab. What we do have to know is the, let's see, what is this? Like what kind of cells are these? Good, photoreceptors, good. Rods and cones, good. Which ones are these? Those are cones. These? Rods, good. Yeah, there's three types of cones. RGB, so red, green, blue. All right, what about this layer?
That's the neurophotoreceptor. I don't know.
Here, let me, so it's like there's this layer of nuclei, and then there's this layer of nuclei, and then there's this layer of nuclei. and this
So
is
outer
inner.
segment layer.
Okay, good. So it's called the outer nuclear layer, because it's,
Oh, nuclei, okay.
mm, okay, what about, this is the outer nuclear layer. Which layer is this?
This is the inner nuclear layer.
Good, the inner nuclear layer. Which layer is this?
This is gonna be the inner nuclear ganglion cells.
nuclei.
Good, the nuclei of ganglion cells. They call it the ganglion cell layer. Just do what it says in the lab manual. Good. The nuclei of ganglion cells. Good. So there's like 3 rows of nuclei, and let's do these next, right? There's 2 places where there are synapses. So the photoreceptors synapse with these ones. What are these ones? Good, bipolar cells. So they synapse with the bipolar cells here. What is this layer?
It's the outer plexiform.
Good, the outer plexiform layer. Plexiform just means network, right? Because that's what they're doing, right? It's like here's a photoreceptor. This is like the dendrite. That's the axon. And then here's the bipolar cell where that's its dendrite, and then there's its axon. And the dendrite, I chose this one. Let me choose this one instead. The dendrite and the axon, they form a network, a plexiform. So this is the outer plexiform layer. What about this one?
Inner plexiform.
Good, the inner plexiform layer, which is the synapses between the axons of the bipolar cells and the dendrites of which cells?
Of the ganglion cells.
Of the ganglion cells. Good. Okay, we did these ones already. Okay, good. And then what is this? Good, the choroid. Good. All right, good job. All right.
Retina, pigmented layer, and then
Sorry, say it again. Oh yeah, retina, pigmented layer, and then choroid. Yes. Good. Yes.
choroid, that's the one that has all the vessels.
The choroid is the one that has what?
All the vessels.
All the vessels. Yes. Good. Hmm?
Where's the outer segment?
Oh, the outer segment.
Yeah, the outer segment.
That's. I don't think that's in the lab Is that in the lab manual?
Yeah.
The
Oh, it
outer
is?
segment, it would be the,
Oh, okay.
the number 2 right in there.
I don't know why it's not on here. This is outer segment, right here. This layer of like the dendrites of the photoreceptors. Oh, is that?
Is that the, like, second layer?
No, it's not that. What is?
Outer segment. It's just called
Sorry.
the outer segment.
So it's the dendrites of the rods and cones.
Mm-hmm.
It says rods and cone photoreceptor segments.
Yeah, it's not on that one. Is it on this one?
Oh, that's,
I think so. Okay, here, let's do it again. So,
this is not histology, is it?
this is histology, yes.
An actual picture?
No, not an actual picture. This is like a drawing. Both of these are like drawings, representations, yes. But it's the same thing, right? Like those are nuclei, all these are nuclei, and then synapses, and then like the dendrites, right? So what is this layer?
Would that be outer segment?
That's the outer segment. It says processes of photoreceptor cells, but it's the same thing as the outer segment, right? Okay, what about this layer?
Outer nuclear layer.
Good, the outer nuclear layer. Let's just do both of them. They have different names for them, it's fine. There's lots of ways to describe these layers.
There is
What-
one in the brackets, outer nuclear.
Sorry, say it again.
There is in the brackets, outer.
Oh, outer nuclear layer, yes. But over here, they call them first neurons, photoreceptors. It's fine. Okay, what about this layer?
The inner.
Good, the inner nuclear layer, good. This layer?
The
Good. The nuclei of the ganglion cells. Good. Same thing over here. And then what about the synapses between photoreceptors and good outer plexiform layer? And then what about synapses between photoreceptor or bipolar and ganglion cells? Good. The inner plexiform layer. I don't know why that was red. Good. What was this again?
That's supposed to be
Right, good. This is the pigmented layer of the retina. And then what about this? That's the choroid. Good.
I don't know why it's so hard for me to see.
So same thing on this side. Okay, just uncover everything. Okay. So yeah, this is how it works, right? The light comes in, and then it hits like here, the outer segments, and then the phototransduction occurs, and then that's what sends the signal up, and then into the axons of the ganglion cells, and then into the optic nerve.
So there's only 2 synapses?
There's,
Oh, there's a lot.
3 synapses?
there's only 2 synapses that we're talking about, like here in the retina, yes. Because, well, like these axons are going to synapse where eventually? Like vision, where does vision have to go through? It's not olfaction, so it must go through the what? The thalamus, good. So like the next synapse, like these axons are going to next synapse onto like,
And they count as in though, right? And some of them go lateral.
sure, but
And
like
then
where do they
they
synapse
synapse
next?
to the thalamus.
hmm
And then through the thalamus, they'll go up. To the visual
Um,
cortex.
So not up, but like back?
Well, back, I'm sorry, visual cortex is occipital,
Exactly, good. Or,
or,
yeah, where's another place that the visual information can go?
oh, so that big colliculi?
Right, which colliculi?
I wanna say superior.
Good, the superior colliculi, nice, good. And I don't remember if it has to go through the thalamus first or if these can just project directly onto the superior colliculus. But for like vision that we become conscious of, it has to go through the thalamus first. So, like that's the next synapse. So there's only 2 synapses here in the retina, but like the next one is thalamus, and then the next one is in the occipital lobe at primary visual cortex. Mhm.
And some of them probably have to association,
Yeah, also association areas, yes. Good. Okay.
or is it a foveus centralis?
Okay, good, right? This is fovea centralis, good. So this is what it looks like, right? So let's name these layers again. What is this?
This is the outer
Good, good. The outer nuclear layer. Here, inner nuclear layer. Here, ganglion nuclei cell layer. Whatever it says in the lab manual, there's lots of ways to describe it. Nuclei of ganglion cells, yes, good. And then it's not on here, but let's just name them. Like, what is this?
The segmented layer, outer segment,
Good. Outer segment here.
outer plexiform,
Good. Outer plexiform layer here.
inner plexiform.
Inner plexiform layer. Good. So notice that. Oh, here. Yeah, that's because there's less synapses, right? Because there's less of these like bipolar cells. Yeah, there's no bipolar cells here or ganglion cells because the light is impeded by those layers. So this is why the fovea centralis has the highest resolution central color vision, right? is because there's no ganglion cell layers, there's no internuclear layer, these axons are coming from like the photoreceptors, and I don't know actually like if they go up and then synapse, but the point is there's less stuff here, so the light comes in and then can hit the outer segments and then cause the impulse to travel, right? So this is fovea centralis. What is this? Good, the choroid. And this one? Good, the pigmented layer. Good. All right.
Of the retina, right?
Of the retina. Mm-hmm.
So one more time, the retina has two layers, right?
Mm-hmm.
One of them will be the neural layer and the pigmented layer.
Exactly. Good. Yeah, this is like the part that's responsible for the phototransduction. And then this is the part that's responsible for like providing metabolic support to the photoreceptors and also absorbing excess light. Mm-hmm. Okay. Fundoscopy. Is that what it's called? A fundoscopy, when they're looking into the eye. So this is what like doctors see. Like this is what they're looking for, right? They're looking for all of these blood vessels coming out of the what? Good, the optic disc, good. And this is more medial, right? Because the optic nerve is more medial. This is more lateral. What is this structure?
This is the
Macula lutea.
Good, the macula lutea, good. And then at the very center, good, fovea centralis, good, good. Okay. Yeah, this is just the same thing, right? So like they have like their ophthalmoscope and then they're looking in and then this is what they see. Nasal, more medial, temporal is more lateral. What is this again? Good, the optic disc. And then this whole region, macula lutea, which means yellow spot. And then the very center of the macula, fovea centralis. Good. All right. So that's vision. Let's do hearing. I guess hearing and equilibrium. All right, so here's the ear. The ear has three parts. What is this part? Good, the outer or external ear. What about this part? Good, the middle ear. What about, good, the inner or internal ear? Good. The outer ear has this thing. What is this? Good, the auricle. What's another name for it? Ear. It's also called the pinna. You might, pinna. Okay, so just do auricle then. Okay. What's the function of the auricle? Auricle?
direct the sound wave.
Good. To like collect and direct the sound waves to the what?
Acoustic, external acoustic canal.
Good. There's a lot of names for it. External acoustic canal, meatus. Acoustic can be auditory instead. Yes. Okay. Good. And what is this?
Eardrum.
Good. Good. The eardrum or the tympanic membrane. Good. Here, malleus. The next one.
Incus.
Good. And the last one. Stapes, good. What's the function of the auditory ossicles?
There's one, amplify the vibration of the sound.
Good, right? So to amplify the sound waves, good. I think they can amplify the sound waves like up to 60-fold, something like that, right? Yeah, without the amplification of the auditory ossicles, you couldn't hear anything, I suppose. Or you could hear like way fewer things. All right, what about, yeah, what are we gonna do next? What is this?
Oval window.
Good, the oval window. Okay, which space is this?
That was a canal.
Internal auditory.
So the whole thing, like all of this is the middle ear, but, sorry, like all of this is the middle ear, but there's really like two parts. So like what is this upper part? It's called the tympanic cavity. And I don't think we learned about the other part, the name of it, but the other part has this thing in it. What is that thing? Good, the round window. Okay, good. What are these?
This is semicircle canals.
Good, semicircular canals. What is this?
This is the station tube.
Good, eustachian tube, auditory tube, pharyngo, tympanic tube. There's a lot of names for it. Good. Okay, what does this tube do?
That connects to nasopharynx. Oh, it helps to equalize the pressure.
Exactly, good. Yeah, so it connects the tympanic cavity to the nasopharynx to help equalize the pressure between the tympanic cavity and like the atmosphere, or the pressure here in the external acoustic meatus. Like have your ears ever popped, like when you go to high altitudes? Well, that's what's going on, is that the like pharyngo-tympanic tube is opening up to equalize the pressure, because when you go to high altitudes, here, let me try to draw it, there's like less pressure. Like, these are air molecules. There's 3 in there, and there's like 5 in there. So the air is pushing this way, so like your eardrum is like bulging outwards, but then you yawn, and the air molecules here, they leave, because the air in here should match the air out here, and when that happens, you like hear and feel that pop, and that is the equalization of the pressure between the tympanic cavity and the atmosphere, right? Yeah, and you need that equalized pressure, otherwise the tympanic membrane is like too taut and it doesn't move as well, right? So that's what the auditory tube does. Eustachian tube, pharyngo, tympanic tube, all right? What is this?
Cochlea.
Good, the cochlea. Here, good, the vestibule, good. The semicircular canals and the cochlea and the vestibule make which structure? Not the internal ear, but there's 2 labyrinths. It's called the bony labyrinth. So the point is, there's this like large bony structure, one continuous bony structure, and it's called the bony labyrinth. And then inside the bony labyrinth, there's another like structure, but it's made of membranes, so it's called the membranous labyrinth. So the bony labyrinth is these 3 parts: the semicircular canals, the vestibule, and the cochlea. Okay. Which nerve is this one? Right, so this one is specifically the cochlear branch. What about this one? Good, the vestibular branch. And then both of those make the which nerve? Good, the vestibulocochlear nerve, the 8th cranial nerve. Good. Yeah, so this one is for sound. This one is for what? Equilibrium. Good. What is equilibrium? Like balance, like balance of the right. Yeah, it's like balance of the head. Like, where is the head in space? You know, is the head not moving, or is it moving? And if it's moving, is it rotating, or is it moving linearly, or is it static? and level, or is it like static and tilted, right? So all of those different head positions, that's what equilibrium is. Okay, and then, I think we did this one. Yeah, it's called the bony labyrinth. So the bony labyrinth has the semicircular canals, the vestibule, and the cochlea.
So the vestibule's like the neck of the snail?
Yeah. If this is the neck, then the semicircular canals would be like the heads, like a three-headed snail. Yeah. Yes.
Three-headed snail.
Yes. Yes.
Terrifying.
So how would we know in like a test, in a lab question, if they're referring to like the semicircular canals or like the bony labyrinth?
I think I would say like be specific.
Yeah, probably like be specific or
And then they'd say like, what's this general structure?
yeah, yep, yeah,
So if it's, like,
specific
this
versus general.
structure, it'd be the bony labyrinth, and
Yeah.
then if it's like, what's this specific structure, it'd probably be like the semicircular.
Mm,
Okay.
yes. Okay, good.
There's one handwriting on these tests that's specifically tricky, and I really have to take a second to look at those. Are the teachers like all write the questions?
Yeah, yeah, just do your best with the handwriting.
I don't think it's your handwriting though.
It's not my handwriting.
Can you write some of the questions?
No.
Sorry. Did you write the practice test? Oh, come on.
Not this semester. Yeah, not this semester. Mm.
But the practice test was you read?
Oh, which practice test?
The one that is out there right now. That
Oh no, that's one of the pals. One of the pals wrote that. Yeah. Mm. Yes. I looked over it, but I didn't write it. Mm. Okay, let's see what else is on here. Yeah, let's skip this actually and go like into the cochlea actually. Maybe we can come back to that. So here's just like a diagram, and then we'll do actual like histological pictures after this. So what is this space? Good, the vestibular duct. It's also called scala vestibuli. What about this one? Good, the cochlear duct. It's also called scala media. What about this one?
The tympanic duct.
Good, the tympanic duct. It's also called scala tympani. Okay. What kind of fluid is in here? Good, perilymph. What kind of fluid is in here? Endolymph. Good. What is this? Good, the vestibular membrane. What is this? Good, the basilar membrane. What is this? Good, the tectorial membrane. What is this representing? Good, the spiral organ, or the organ of Corti. What is this?
This is, isn't it the axons?
Mm?
Hair cells?
Yes.
They are attached to hair cells, but they're axons for the
So which nerve?
cochlear branch of the vestibulocochlear nerve.
Good. And then what is this?
Spiral ganglion.
Good, the spiral ganglion. Nice, good. All right, let's do it again. Which space is this?
Vestibular duct.
Good, the vestibular duct. What about this one?
The tympanic duct.
Good, the tympanic duct. This one?
Cochlear duct.
Good, the cochlear duct. What is this?
The vestibular membrane.
Good, the vestibular membrane. What about this? Good, tectorial membrane. Good. What is this whole structure?
Spiral organ.
Good, the spiral organ. What are these cells of the spiral organ?
Hair cells.
Good, hair cells. What is this over here?
The spiral ganglion.
Good, the spiral ganglion. Good. Okay, good job.
What's the top right?
Oh. Top right.
Yeah,
Here?
no, oh, just kidding.
It just says ID.
I just, yeah, it's on the ID, but it's referring to the vestibular membrane?
Mm.
Okay.
Okay, good. Okay, let's do this one. What is this? Good, the malleus. Oh, this one is describing like how is the sound actually transduced. So let's go through some of that. What is this?
Incus.
Good, the incus. Here,
Stapes.
good, the stapes. Okay, so there's some pressure wave. The pressure wave is oscillating the tympanic membrane, which causes all of these to oscillate. And then the stapes is pressing on which structure? Good, the oval window. And the oval window is pressing on which fluid? Good, the perilymph inside of which space?
The vestibular duct.
Good, the vestibular duct. Oops, don't look. So that's what all of this is. That's the vestibular duct, or scala vestibuli. And then depending on the frequency of the oscillation, it'll deflect the basilar membrane in certain locations. So if it's high frequency, like where is that detected? Like which part of the basilar membrane is deflected when the high frequency vibrations come in? So-
Would that be high frequency closer to the oval window?
Exactly. Good.
And the low would be further away from the oval window.
Exactly. Good. Good job. So, so like here, maybe I'll just write like this is for high frequency, and then over here is for low frequency, right? Because closer towards the windows, the basilar membrane is narrow and stiff, so you need a lot of energy to deflect it, and so the brain basically interprets that as high frequency. It is high frequency, right? Versus low frequency vibrations, they don't have enough energy to deflect this portion of the basilar membrane, so it has to travel through the scala vestibuli before it can cause the deflection of the basilar membrane. So it'll deflect, and then that puts pressure in the perilymph in which space?
Perilymph in tympanic.
Good, in the tympanic duct or the scala tympani. And then the perilymph goes this way and then bulges on what? On the round window. Good. So like it bulges like out, and then it goes back in. And then when it goes back in, it goes back to like that same place. And then once it goes back to that same place where it deflected, here, we have to go to another one actually. Let's go to this one here. So it went down, and then it goes back up. And then when it goes back up, these, what are these called?
Hair cells. Stereocilious.
Right, good. So these are the stereocilia of the hair cells, and it's really only these hair cells that produce the sound. So when the stereocilia deflect, which ion rushes into the hair cells? Like, did you have to learn about like endolymph is rich in which ion? It's not sodium.
Potassium?
Good, potassium. Good. This is getting more into physiology, but let's just keep going, right? So potassium goes into the hair cell, and then the hair cell releases neurotransmitter onto the dendrites of these bipolar cells, and then it goes through the spiral ganglion, and then like where are some final destinations in the brain where this auditory signal can end up? Like name 2 locations.
Inferior
Inferior
canicula.
colliculi,
And temporal lobe.
and the temporal lobe. Good, right? Because the temporal lobe has the auditory cortices, right? Primary auditory cortex. Right, there's like an association area, like kind of there, like between the temporal lobe and the parietal lobe. Yes, Wernicke's area, exactly, right? Assigning meaning to the sounds, right? It's not just some random sound. It's like, oh, that sound means like dog, or that sound means like, what's the meaning behind the sound? Yes, good. It sounds like what?
That sounds like Tiger Run.
Like Tiger
Well,
Run?
Because when you have association, it also evaluates for threat. Like if you hear a kitten, you're not threatened by that, right?
Mhm.
If you're hiking in Helen Puddin Park
Mhm.
and you hear a big cat yawn,
Mm.
you're gonna take off.
Yes.
Or do something. So there's gonna be like flight or fight reflex at that point.
Yes.
But it evaluates all the sounds to see whether or not it's the threat.
Yes, exactly. Good. Yeah, we have like a whole library of sounds in our temporal lobe and some of our association areas that our brain is comparing like this sound that just came into. So it's doing exactly that, right? Where it's like, is that a cat? It's like, what kind of cat? Like a small cat or a big cat, right? Like if it's like a kitten, it's fine. But if it's a lion, it's not fine, right? Yep. Good. Okay. So all of that was cochlea. Let's talk about the vestibular apparatus.
No, this is
Um,
this is the ampulla.
Good, the ampulla. So this is the ampulla of some semicircular canal and duct. Good. Okay, so that's what this is, ampulla. Ampulla is like a swollen thing. What is this?
Crista ampullaris.
It's not the crista ampullaris, no. What is this gel?
No, we don't have to know that.
Oh, do we not have to know it for, oops, it's not in the lab manual? I don't know why it's on.
I don't think we particularly don't have to know that. I
Okay.
don't know if anybody else do.
Okay.
I wrote it for lecture.
Okay, for lecture, maybe you do. Can we just go through it? Because it's like to understand how the semicircular ducts work, I think you have to understand like what's inside the ampulla, right? Like how do you know crista ampullaris? Is that in the lab manual? Or is that a lecture thing?
Lab manual.
Oh, okay.
It says ampulla with crista ampullaris.
Oh, with crista ampullaris. Okay, got it, got it. Okay, so maybe I'll give you some extra information or information that you only need to know for lecture, but let's go through it because I think it's useful to know how does your head detect angular acceleration? Well, it does that with this thing. It's like this gelatinous substance. It's called the cupula. And the cupula is sitting on this, which is like a specialized type of epithelium called the crista ampullaris. There's different cells of the crista ampullaris. I don't know where, oh, there it is.
What's the Krista Ampullaris supposed to do?
That's the bulging out thing, right?
Yeah, sorry, let me finish it. So it's like, there's like this crest, and on the crest, there's all these different types of cells. The sensory cells are the ones that have these long stereocilia inside of this gel, and when your head rotates, there's fluid in here. What's the fluid in here? Good, perilymph. The perilymph has inertia, so it doesn't want to move when your head moves. Like this is your head. All this is connected to your head. So your head goes this way, right, if you're rotating it. But then the fluid doesn't want to move, so it like runs into the gel, right? So then when it runs into the gel, it causes the gel to bend, and then when it bends, it causes the channels within the stereocilia to open, and then that's what causes the influx of potassium into the cell, and that's what sends the signal through the vestibular nerve into your brain, which lets you know that your head is rotating, right? Sorry, this is not perilymph, but endolymph then,
Oh, yeah, it's in the dump, right?
right?
Because perilymph would be outside.
Exactly.
Okay, sorry.
Good. Okay, so that's how it works. I don't know if that's helpful or not, but in lecture, you'll probably learn more about how that works specifically. Okay. And well, it's 10:55, and we got through special senses, and I don't think we'll have enough time to get through anything else. So that's all that I have. Yep. Are there any last questions? What's that?
Do you have the linear movement?
I don't. Yeah, I don't know where that one is. Hey, let's just talk about that though, right? Maybe let's just talk about it because I don't have any pictures for it. So
The linear movements, so when we're talking about the macula, right?
mhm.
Macula is different than this, it's basically, if I'm not wrong, hair cells sitting in gel
Mhm.
with, like calcium crystals inside.
Mhm.
And, when you tilt your head, that gel basically slides down because of that crystal's weight, and you feel it, well, that's how the action potential fires that your head is tilted.
Yes. Yeah. Let me try to find something actually to put a visual to that.
The macula with the utricle and the sacral?
Yes.
I think it's before that.
It's before this one.
Yeah, I think right here.
Okay. Oh. So yeah. So. Yes. So this is what's inside of the utricle and the saccule. It's the same thing basically, right? It's like a bunch of cells, the like sensory cells and the supporting cells, and then like some gel sitting on top of that, and then the sensory cells have the stereocilia sticking up into the gel, right? So the structure of the utricle and the saccule is like the same as the ampulla. So when your head is upright, there is no bending of the stereocilia, but if your head is tilted, then this otolithic membrane with all of these, like all those dots are otoliths, which are like calcium carbonate crystals. They provide weight to the gel, and the gel causes the stereocilia to bend. And then when they bend, that causes the potassium to rush in, which causes the depolarization of the cell. And then that's how your brain knows that your head is tilted, right? Versus like it being static. And this also is happening for linear acceleration. Between the utricle and the saccule, which one detects horizontal linear acceleration?
Utricle?
Good. Which one detects vertical linear acceleration? The saccule. Good.
Can you repeat that, the horizontal, which one?
Yeah. So the function of the utricle is to detect horizontal linear acceleration. So like moving like forwards, backwards, side to side, and like anything in between those two axes. It's detecting acceleration in those planes, right? So like if you're in a car, that's accelerating, or if you're in anything at all, right? Like moving fast, a jet, I don't know, going fast, right? Versus like the saccule is for vertical linear acceleration. So like if you're in an elevator that's falling, or like one of those rides at Disneyland, Disney World, like the Magic Mountain ride. Is that what it is? I've never been on one of those. But it's like if you're going up really fast or down really fast, that's what the saccule is detecting. And if you're going forwards, backwards, or side to side, or anything in between really fast, that's what the utricle is detecting, right? And then the semicircular ducts are detecting like not linear acceleration, but like angular acceleration, like rotational, right? Because that covers all of the ways that objects can move in 3D space, right? That's what your vestibular apparatus is doing, is detecting like, is your head like moving? Is it accelerating, right? Because that's different than just velocity. like if you're in a car going a constant velocity and your eyes are closed, you don't know if you're moving or not, right? You only know if you're moving if your velocity is changing, and change in velocity is acceleration, right? Have you guys taken physics yet? Or do you have to take physics at all? Okay. They talk about these things in physics, the difference between like position, velocity, acceleration. So it's like that's what your vestibular apparatus is doing, detecting all of these like ways that your head can move in space. Okay.